Hello, everyone, and welcome to today's webinar. My name is Olga Bodie, and I'm part of the team here at Syngap Research Fund. Our presentation today is SRF, or Syngap Research Fund, and our 5 years of funding innovative research. I have the pleasure to introduce today's speaker, Dr. James Goss. Dr. Goss is a neuroscientist with over 30 years of experience in basic and translational research, including broad involvement with drug development in academia, biotech, pharmaceutical, and nonprofit organizations. Dr. Goss has overseen the preclinical development of gene therapy, biologic, pharmaceutical, and device-based therapeutics. He currently serves as a scientific director of the STXVP1 Foundation. Excuse me. A recorded version of this webinar will be available on the SRF website under webinars. And by the end of this presentation, you will have the opportunity to get answers to your questions. We'd love to hear from you. So please write those questions in the Q&A below. For those of you just joining us, welcome. And again, our speaker today is Dr. James Goss. His presentation is SRF and our 5 years of funding innovative research. It's now my pleasure to turn things over to Dr. Goss. Thank you.
Thank you, Olga, for that nice introduction. Let me see if I can share my screen. And let me know when you can see my slides.
Yeah, we can see. It looks good.
Yep. All right, thanks very much. So, yeah, the title of my talk is 5 years of funding innovative research for Syngap1 and what to expect moving forward. So I just don't want to focus on the last 5 years. I'm going to also talk about what to expect going forward. In fact, my outline for my talk today is the first thing I want to talk about is what has the funding over the last 5 years achieved. And then what I want to do is I kind of want to move to the future and kind of summarize and review different therapeutic approaches that are coming in the future. And then I want to kind of finish with discussing clinical drug trial challenges that I think everyone should be aware of when it comes time to do clinical trials for Syngap1. So what has 5 years of funding achieved? Well, if I start off at a high level, close to 4 dozen grants have been funded through SRF. This is over $4 million worth of funding that has been provided or committed at this point. Over 30 funded scientists at over 25 institutions or organizations. And this has resulted in several publications. I don't know the exact number at the moment, but it is at least half a dozen and growing on a yearly basis. So there are 4 main avenues of, I think, what SRF funding has achieved that I want to discuss in a little bit more detail. Those are, the first is increase the number of scientists who are working in Syngap1. And the second is this funding has developed research tools for Syngap1. The third is it has started therapeutic development for Syngap1. And the fourth is it increased clinical trial readiness for Syngap1. So first off, increasing the number of scientists working on Syngap1 has been a very important achievement in this funding. So back when SRF was first started, the number of scientists who are actually working on Syngap1 was very small, just a few scientists, and no real drug development was being done in Syngap1. And funding has changed all of that, and the funding specifically provided by SRF has done that. And how does that do that? Well, easy. So when you fund research, you're bringing in more scientists to work in a field. So more researchers means more knowledge in that field has been generated. And so as the number of grants that are funded by scientists grow, the amount that we know about Syngap1 grows, both in its basic biology, its basic function, and its basic dysfunction that occurs within Syngap1 disorder. All of that knowledge grows. This additional insights into the functions or the of Syngap then leads to what we can refer to as new drug targets. And these are, so when developing therapies target something within the biology of a disease process. And the more we know about Syngap1 function, the more targets become available that can be druggable for to develop new drugs. Any new drug or any drug approach that is developed from this knowledge are additional shots on goal for finding therapies that are going to work in Syngap1. Let me give you 3 recent examples. And these are 3 investigators who were funded in this last round of funding through SRF. Helen Wilsey, who's at University of California, San Francisco. She was funded for a program where she's looking at the role of Syngap1 in neuronal cilia. Neuronal cilia is a structure in neurons. Most cells have cilia, these cilia structures in them. Not a lot is known about what neuronal cilia do. But what she has found is that Syngap1 is expressed within neuronal cilia, and she was funded by SRF to get a better understanding of what SRF is actually doing in neuronal cilia. If her research is successful, basically a target that she can find are drugs that reverse cilia defects. Her hypothesis is that neuronal cilia are defective in Syngap1, and that if you reverse that defect in the cilia, then you may have some beneficial effects in the disease process. And this is an additional shot on goal. Baptiste Lacoste at the University of Ottawa was also funded. And what he's looking at is the role of Syngap1 in neurovascular development, or basically how blood vessels develop within the brain. And he is suspecting that the Syngap1 plays a role in this. In the Syngap1 disorder, basically there may be a problem in the neurovascular. And this leads to some of the problems associated and that we see in Syngap1 patients. That might be able to be targeted by looking at vascular drugs. So drugs that instead of are affecting the neurons are affecting the vasculature of the brain and the blood vessels. Again, this is an additional shot on goal. And third, Paul Donlan-Asp, who's at the University of Edinburgh, is looking at the regulation of Syngap1 expression in neuronal dendrites. He's specifically looking at the effects of what are called RNA binding proteins on how Syngap1 is expressed. And by targeting RNA binding proteins with specific sorts of drugs to make them either work better or increase the expression of Syngap1, then that is another potential way of achieving a therapeutic effect in Syngap1 patients. So yet another additional shot on goal. And so bringing all these investigators into the Syngap1 field is improving our knowledge of Syngap1, and it's going to lead to better and newer therapeutics. A second important achievement for the funding that SRF has provided is to develop research tools for Syngap1. So these include many different things, but some examples here are iPSC patient and control cell lines that have been developed under SRF funding. In these examples on this slide and in the next slides, the parentheses are labs or organizations that have been funded by SRF who are doing the work. Syngap1 mouse models, there are several mouse models available, and SRF has funded some of those mouse model developing. There's a rat model that's being developed, and there's a frog model of Syngap1 that has been developed. All of these models help scientists better understand how Syngap1 functions, and they're importantly used for translational research. And basically what that means is taking kind of the basic knowledge that we understand about Syngap and using that to try to find, as I said before, druggable targets and testing potential drugs. So all of these models, so iPSC cell lines, mouse models, rat models, frog models can be useful in testing drugs. And so this is a very important resource to have, and the foundation has done an excellent job in funding research to develop many of these resources. The third are developing of various therapeutics. And so SRF has kind of in the beginning of funding had funded primarily a lot of basic research, but now more funding is moving towards more applied research or therapeutic development. And there are several examples of this that have been funded by the foundation. So first off is drug repurposing. And so drug repurposing is taking a drug that has already been approved by a regulatory agency like the FDA that's already for sale out there and marketed, but was not originally intended to treat Syngap1, and using it to see if it can in fact treat Syngap1 or at least some of the symptoms associated with Syngap1. And so one example is 4-phenylbutyrate, or Ravicti, and I believe that the first Syngap1 patients have been treated now by Zachary Grinspan at Weill Cornell with 4-phenylbutyrate. 4-phenylbutyrate is a drug that was originally developed to treat a urea cycle disorder. It had nothing to do with the brain, has nothing to do with neurodevelopment disorders, and nothing to do with Syngap1. But it's possible. Basic research had found that phenylbutyrate may actually have a protein stabilizing effect, and had been tested. And because of that, we're tested in a couple other neurodevelopmental disorders, and now is being tested in Syngap1. And so that's a perfect example of drug repurposing. The foundation has also supported large-scale drug screens. This is, instead of just taking one drug and testing it, what a large-scale drug screen is, you develop a way of taking a lot of FDA-approved drugs and see whether or not they have any effect on Syngap1, for example. So in the case of Rarebase, this is a company that the SRF funded a study with. They took something like 3,000 to 4,000 different drugs and tested to see whether any of those drugs could increase Syngap1 expression in neurons. And so since this is a large screen, this is a stepwise procedure. You first test all of these drugs, you see what works, and then you do more validating experiments beyond that. But the idea is eventually you can identify several drug candidates that you think may have the ability to have some effect on Syngap1. If you can increase expression, then perhaps these drugs, again, which are already FDA approved, might then be able to be tested in Syngap1 patients fairly quickly. A similar drug screen was done in the Chow lab using flies. And then there are some small molecule drugs that are being developed in different labs. And so a small molecule drug is basically the type of drug that almost everybody is familiar with. So if you're taking a pill for anything, that's a small molecule drug. And these are basic pharmaceuticals. And there are labs that looking at drugs that are structural stabilizing drugs. Basically, this just means that they help proteins stop being degraded in cells, for example. And this is a way of trying to keep the Syngap1 that's made in a cell or in a neuron around longer. There's another group that are testing drugs that affect a potassium chloride protein in neurons with the idea of trying to rebalance the excitation and inhibition within the brain. And so these small molecule drugs, they're not FDA approved, but nonetheless, these are all potential shots on goal in potentially developing. Some of these drugs may move forward in clinical drug testing, depending upon how the studies that the foundation have supported turn out. Other classes of therapeutics that the foundation has been supported are what we call precision therapeutics. As opposed to what I talked about in the last slide, those are drugs that can affect not specifically Syngap maybe, but affect neuronal function as a whole. Precision therapeutics, on the other hand, target Syngap specifically, and there are a number of different precision therapeutics that are being looked at for Syngap1. So these include antisense oligonucleotides, or ASOs, that you may have heard of. I'll talk more about these in a few slides. There is an RNA booster, which is a precision therapeutic aimed at trying to increase the amount of Syngap1 RNA that is made, and the amount of Syngap1 expressed from RNA. There is gene replacement of Syngap1 that's being looked at in the Cobb and Kine lab, in the Zemplenny lab. Gene activation and cell therapy are types of precision therapeutics that are being looked at, being investigated by people who have been funded by SRF. And as I said, I will discuss these in a little bit more detail. The fourth major achievement that the funding has brought us is increased clinical trial readiness. And this is very important for the future. And so basically what I mean by this is that you're getting, by funding the work, by funding specific studies, the foundation is getting the Syngap1 population ready to go into clinical trials and making Syngap1 an attractive disease for pharmaceutical companies to develop drugs for, because increasing clinical trial readiness does, and I'll discuss here in a little bit more detail, is that it de-risks Syngap1 as a disease for pharma to develop drugs for. So developing drugs is very expensive, and drug companies want to make sure that they spend their money wisely, and they go after diseases where they have the highest likelihood of finding a a positive effect of their drugs and will be able to get a drug approved. And so making your population attractive to pharma companies to develop drugs is very important. And so how has the foundation done that? Well, by funding companies like Probably Genetic to identify more Syngap1 patients. So the more patients that there are available, the more likely pharma company is going to develop drugs for. Better understanding variants of unknown significance. So Syngap is a disorder that has a lot of VUSs, and these are variants or mutations in the gene where it's really unknown whether or not the mutation is pathologic or not pathologic. Being able to understand what VUSs are pathologic is very important because if you have VUSs where you're not really sure whether or not the mutation has a pathologic effect or what the mutation does in Syngap, then drug companies are going to be less likely to utilize patients who have those particular mutations in drug trials. Defining the natural history of Syngap1 disorder is very important, and natural history is basically how the disease naturally progresses over time, you get to understand this through a natural history studies. CHOP is doing something like that for Syngap1 now, but you also understand this through a disease concept model, which is basically understanding what are the major implications of Syngap1 on the patient and on the family and caregivers. And the foundation has funded studies in disease concept model in trying to understand the natural history by using citizen data, for example. Another important thing for clinical trial readiness is understanding what are called outcome measures. These are the actual things that are looked at in trying to determine whether or not a drug, for example, is having an effect on a disease. And so you have to have something that you can measure in the population and see that that changes in response to a therapeutic intervention. So the foundation has funded some studies that are looking at different sorts of outcome measures. There's a neurobehavioral evaluation tool that is being developed. EEG biomarkers are being developed to help as a potential outcome measure, and improving the seizure diagnosis, specifically from caregivers, how caregivers view and look at seizures and be able to diagnose when their child is having a seizure. So these are all very important, all things that the foundation has funded. So I now kind of want to move from what the foundation has funded to having kind of a overlook of, so where are we going now that you funded these studies? We've seen what the funding has achieved. Where is that taking us as far as what the real goal is, which are therapies for Syngap1 and Syngapians. And so we can kind of look at therapeutic development in different types of therapies. So we talked about repurposed drugs. Repurposed drugs are currently in now, and as I said, with the 4-phenylbutyrate trial, But between now and 3 years, we're going to see more repurposed drugs, I think, in the Syngap field from the studies that the foundation has funded, as I spoke before. And so this is something that is coming soon, and so we want to be ready for that. Kind of the next types of drugs that are coming after repurposed drugs are the antisense oligonucleotides. And I would think that within the next 2 to 5 years, you will see some ASO drugs being tested in Syngap1 patients. And then following that are what we call gene therapies, or what I will call gene therapies, in 3 to 7 year range. I would say you start to see some of the initial gene therapy trials in Syngap1 patients. So let me talk a little bit more about drug repurposing and why we go after drug repurposing, and why it is something that's exciting. So the best way to discuss, I think, drug repurposing is to compare traditional drug development with repurposed drug development. And so on the left, what we see here is what traditional drug development how this works. And so basically, a scientist has an idea. They have what we call a target or a disease-based research, i.e. they have an idea that they think, I want to treat some disease. What should I go after in that disease? They do some basic research in that disease, and they get what are called lead compounds, or these are, here's a set of potential drugs that might work in the disease. And we will do more testing of those lead compounds, and that testing, at the end of that testing, you get a candidate drug. And this is the best compound that you have that you think is going to have an effect on your disease. And then that candidate drug goes through what's called preclinical development. And this is additional work on this drug before it goes into people. And preclinical development involves a lot of different steps, but part of that some of that are things like toxicology. Is the drug toxic to animals? So you want to know if that's the case before you give it to people. And then you also do some additional animal studies to see what happens to the drug when you give it, other than toxicology. Is it toxic, but is it actually working in a disease model in an animal? Once you do the preclinical development, then you go into clinical development. Clinical development is you're giving it to people, and there are clinical development can take a while. There are different phase 1, phase 2, phase 3 clinical trials. You've probably heard of those terms. That's all part of clinical development. You test it in people, and then once you get all the data in people, you go through what we call registration. That's going to basically the regulatory agency, the FDA. You're giving them the information that you've collected from all of this that you've done. The FDA determines whether or not the drug is safe and effective. And if it is, they basically registered the drug, and now you can sell it to the market. This takes a long time and costs a lot of money. And it has a high failure rate. Most probably, from target disease-based research to market, there's probably less than 1% of that actually make it to the market. And even at clinical development, from clinical development to market, there's about a 45% failure rate. So even at that advanced stage. So what repurposed drug development does, on the other hand, is you already have drugs which have been clinically approved. They're already marketed. And all you are doing is you're taking those drugs and you're saying, okay, well, they were approved for this condition, but I'm going to use them in my condition. So again, going back to the 4-phenylbutyrate, you're taking a drug that was originally created and marketed for urea cycle disorder, and you're seeing, I think this can have a positive effect in Syngap1. So now you take that drug, you do some screening and validation tests, basically just to make sure that it's having an effect on the target, in this case, Syngap1, that you think it's going to have. If that does, then you still do some clinical development, but the clinical development in this case is you may just take that drug and test it in a handful of patients to see whether or not it has any efficacy. At that point, then you can go into registration and marketing. And this is something that a drug company might end up doing. You don't actually need to take a clinically approved drug and go through registration and marketing. You can just take the clinically approved drug once you demonstrate that it does work in people and it has an effect on the disease, and use it off-label, which basically means that it's not marketed for the disease, your disease. It's not marketed for Syngap1, but you can use the drug off-label. There are some pros and cons to either of those, marketing or off-label use. Ideally, if you go the market route, then basically insurance will cover the use of that drug for the new disease. So now I kind of want to discuss the precision therapies that I discussed in the previous slides. I said I'd talk a little bit more about them. And so I wanted to delve into these in order to give you a better understanding of exactly what we're talking about when you hear terms like gene therapy or ASOs, what exactly is going on, and how are these exactly working. It can be a little bit confusing. And so one type of gene therapy approach is gene replacement. And if we take a look at the picture on the right, we have a cell, we have a neuron here. In our case, you have a defective gene. That gene is within the nucleus, which is that shaded area within the cell. You have that gene undergoes transcription, which creates an RNA. That RNA is translated into a protein. If the gene is defective, it makes a defective RNA. And that RNA, when translated, makes a defective protein. Gene replacement is basically an idea where you take a normal copy of that gene and you add it to the cell, you add it to the neuron. That normal gene then makes normal RNA and normal protein. And this is a basic type of gene therapy. It is, as far as gene therapies are concerned, the most studied type of gene therapy, mostly because it's one of the easiest types of gene therapies to think of. Just adding the normal gene into a cell. It's generally been utilized for monogenic disorders like Syngap1, for example, but can be used to increase the expression of a normal protein, if that's what you want to do. It generally results in overexpression of the gene of interest because it's difficult or can be difficult to control the expression of the gene that you're adding. So it is permanent. So once you add a gene into a cell, in this case, it's going to continue to make that normal gene, and it requires a delivery system. And for the most part, that delivery system is known as a vector, a gene therapy vector. That's what the delivery system is, the thing that actually gets that gene into the neuron or the cell of interest. There is what's called gene manipulation, or instead of adding an extra gene, in this case, you're using what we'll call molecular tools to directly interact with the malfunctioning gene. So we have the same system set up here on the right, but in this case, you're adding a tool that actually goes and repairs, for example, the gene, the mutation in the gene. And there are several types of gene manipulations or gene editing. There's direct gene editing, which is repairing the gene. CRISPR-Cas, you've probably heard of CRISPR-Cas. So that gets in the news a lot. There's another type, a couple other ways of repairing mutations in gene, prime editing, base editing. I won't get into the details of those, but there are several different ways of editing a mutation in a gene and trying to directly fix it. Instead of directly fixing a gene, you can either deactivate the mutant gene, or you can try to activate the non-mutant gene. So yeah, as you know, everybody has 2 copies of each gene. And so you can deactivate the bad copy or activate the good copy. And there are a couple of ways that you can do that. CRISPR-A and CRISPR-I are 2 ways that are being looked at for a lot of genes. And there are other sorts of activation. Zinc finger proteins are one. Again, I won't get into the details for that, but there are a lot of these molecular tools which are available. So again, gene manipulation is very specific to the gene that you're looking at. One of the positives is it maintains cellular control over the expression of the gene, so you don't get overexpression issues that you might get with gene addition. It is technically challenging. It's more technically challenging than just adding a gene. Because basically you're still adding something to the cell, but in this case, the molecular tool has to get into the cell, into the nucleus, find the right gene, interact with the gene, and have its effect. So there's a lot of efficiencies that need to be overcome with that. Like gene replacement, it's permanent. Once you put these molecular tools into cells, they stay and they will work and they will fix the gene, and it will permanently be fixed. And like gene delivery, it also requires a delivery system. And then there are RNA therapies. So in this case, again, we have 2 copies of the gene, your defective gene, the normal gene, making defective RNA and defective protein, or normal RNA and normal protein. In this case, you add something that is going to interact with RNA, ASOs, antisense oligonucleotides, which talked about already. These generally interact with RNAs, and they can do things like either work on the defective RNA and silence it so it doesn't make any protein, or they can work on the normal copy of the RNA and enhance it so it makes more normal protein. There are RNA ASOs that, as I said, you have RNA silencing, that RNAs that block proteins that bind to RNA. By blocking those proteins, you increase the expression of that RNA. You can modulate how RNA splices together in what's called what's called exon skipping. I won't get into the details of that, but There are already drugs on the market, ASOs on the market that do all of these things. And so this is very real technology. And as I said, on the horizon for Syngap. There's also what are called signups. These are basically long pieces of RNA with a attached to an element that enhances the expression of your target RNA. I don't know if anybody's actually using these for Syngap at the moment, but these are being actively investigated for other disorders and could potentially be used for Syngap1. Like replacing a defective gene, you could also replace RNA. So you can just add RNA into a cell and see if and create normal protein from that added RNA. And you can also use what are called read-through drugs for specific types of mutations. So if you have a nonsense mutation or a mutation that basically stops translation from occurring from an RNA, a read-through drug is a small molecule, generally, that can basically allow that translation to proceed through what would be a nonsense mutation. RNA therapies are simpler technology than gene replacement or gene editing. They're cheaper to manufacture than either of those. It's also specific. RNA therapies are transient, so they have to be redosed. So ASOs, even though they're cheaper and simpler, using ASO as a therapy, patients have to be redosed on a regular basis, whether that is currently usually every couple of months or every quarter or every 3 or 4 months, because RNA doesn't last. RNA is degraded normally in cells and so will disappear, and so these have to be redosed. And ASOs or RNA therapies usually also require a delivery system, though not always. You can inject just straight ASOs into certain body fluids. So delivery methods, I don't want to get too much into this, but for if you're going to deliver genes or those gene editing molecules I talked about, oftentimes you need some sort of delivery system to do that. One way to do that are to use what are called viral vectors. These are basically viruses that have been basically taken apart and kind of put back together so that they don't cause disease, but they can be used as a delivery vehicle. And you can think of that as viral vectors as being a truck, and the gene is the cargo that's contained within that truck. And there are several different viral vectors that can be used. The most common are AAV and lentivirus. You've probably heard of AAV. You may have heard of lentivirus, but they all have their positives and negatives. All of these have been utilized as the different types of viral vectors. There are what are called nanoparticles that can be used as opposed to viruses. These are small encapsulating molecules. Some are lipid-based, some are polymeric, and basically they form a protective barrier around whatever it is that you're trying to deliver, whether that is a gene or an RNA or even a protein. And again, they have some positives and negatives associated with them. They're used much less frequently than viral vectors, but there's a lot of research involved in looking at these and trying to develop these as delivery vehicles for various types of precision therapies. And the third are exosomes, and exosomes are basically structures that are already created by cells and released by cells, and they are basically a membrane, like a cellular membrane, that's encapsulating a solution of different compounds. Those compounds can be proteins or nucleic acids, DNAs, RNAs. As I said, they're normally made by cells, normally released by cells, but they can be engineered so that you're making specific sorts of exosomes that contain specific molecules in them. And those can be used for delivering therapeutics. And in fact, the foundation supports work in this by one of the grantees, Zemplenny at the University of Nebraska. Yeah, I want to kind of finish up my talk on discussing a couple of clinical trial challenges that I think everybody needs to kind of keep in mind as we move into precision therapies and start doing ASO clinical trials and gene therapy clinical trials, and also small molecule clinical trials for Syngap1. And so some of the challenges are, obviously, there's a small population of Syngapians, and this is something that you always have to keep in mind. The population isn't large enough to have large clinical trials for most diseases that occur in the general population. When testing a drug, hundreds of people will go through a clinical trial, go through clinical trials to test the drug. We obviously can't do that with Syngap1, so we're limited in trial participation. A given trial will only be able to use a handful of Syngap1 patients, and the population obviously is small and distributed among multiple countries and regions, and so this has to be kept in mind. And this is something that I think the foundation will probably need to will be important for the foundation to understand and to maybe guide patients into what clinical trials to participate in. As I said, and hopefully there's a situation where there are so many good potential therapies that a lot of clinical trials will come up. That would be a great problem to have. We may have to pick and choose which clinical trials would be best to take part in. Another challenge is clinical trial design. So traditional clinical trials have placebo groups, which are a group of people who get a no drug treatment, basically. And this, again, is a difficult thing to do in a rare disease like Syngap. We can't really afford to have traditional placebo groups in clinical trials. But there are ways of clinical trial design that can get around that for things like, for drugs that can be redosed, like small molecules or ASOs. For gene therapies, ideally, you don't want any placebo group, because some gene therapies, like it's a gene therapy using an AAV vector, you may not be able to redose people. So you can't actually give them a placebo, which would be an AAV vector without the Syngap in it, right? So you can't really use that as a placebo group, because that would preclude those patients from getting an active drug later. So this is something that is is being discussed with the FDA, with a lot of different rare disease groups. And we need to find ways around having to use placebo groups. So far, the FDA has been somewhat reluctant to get away from having placebo groups in gene therapy trials. Ideally, but regardless, we have to maximize the amount of data that's obtained from each participant in any clinical trial that's done. And because again, with the small population. The other things to keep in mind are clinical trial outcomes. I've talked about these already, but these clinical outcome measures are difficult for a lot of neurodevelopmental disorders like Syngap1. How do you determine whether or not somebody is improving on a drug? Obviously, if the drug is having an amazing effect, that's easy to determine. But if a drug is having a limited effect, then how you measure your outcomes are going to be very important in getting a drug approved. So you have clinical outcomes which look at multiple domains: communication, movement, cognition, what have you, within the population, and do those approve. The ability to harmonize clinical outcomes between trials, which basically is, if you're going to run a number of clinical trials with different therapeutics, you want to be able to measure the same outcome in all of those trials. And so that's what I'm talking about with harmonization. And the use of biomarkers and the development of biomarkers to help with the clinical outcome measures. Do you see changes in EEG? Do you see changes in some sort of marker that you can see in the blood? And then timing also is important. When do you treat patients? The earlier you treat them, is that the best time to treat them? Can you treat adults? And how long do the clinical trials need to last? So for a lot of clinical trials in most diseases, the clinical trial may last, a couple of months to look for an effect of a drug. But for a neurodevelopmental disorder, it might take a long time between the time when you treat somebody with a drug or therapy and you actually start to see an effect. So clinical trials may need to take longer in the population. And I think I way over talked what I needed to talk, but sorry about that. But I will take some questions if anybody has any.
Hey, Jim, thank you very much. That was super comprehensive.
Yeah, it's a little, sorry.
That's okay. I actually have a couple questions about trials. So first off, I mean, it's so great to see the history of what SRF has funded and new avenues and stuff. So thank you very much for that summary. But you're right that the future is what we're always really interested in. And so, for our first early trials, we're really looking at children who have countable seizures, right? So countable seizures is really what the FDA understands how to deal with, but that's not necessarily our entire population, but it is some of our population. So if we start there and look at that, I'm wondering, what else do you see measuring on a short time scale, and what do you see measuring on a longer time scale? And then can we have sort of like phases where, getting something to market might be on the short-term scale, but then what can we do for, understanding the longer term might end up being what somebody like my own kid, who doesn't have that many seizures, thank you very much, but is very, has severe cognition issues, right? And so that may take longer. And then the final question will be, with development, I might see him getting better, but he might actually be harder in some ways, right? Like, so if you can't let's just say when he was first born, he didn't, attend to the world at all, right? And it took him a really long time to even attend to the world. And so he's a very easy baby. But then as he grew, he got harder and harder, right? So as his development increased, his ability to show distress really increased, right? And so you can imagine someone getting better, getting sort of older, able to do more things, but also getting more frustrated or getting, do you see what I mean? So it's like, from a parent's point of view, you're watching your child, but then there's always new problems, right? But we're happy with it because they're developing. So anyway, I kind of want to talk about that a little, because I feel like some of the scales don't necessarily measure just how low we're starting.
Yeah, no, this is a major issue with all neurodevelopmental disorders. And let me kind of go from the beginning. And yes, you're right. So
Sorry, that was a lot. That's all I have. Yeah.
No, that's fine. Yeah, you're right. I mean, for the first trials, really, you're looking at a seizure frequency, because as you said, the FDA understands that it's something that is objective, that can be counted. And because it can be counted, it's easy to say, okay, did seizure frequency change due to drugs, right? But you bring up a good point. Not all kids have seizures, and depending upon the disorder, and I'm, I can speak to SCXBP1, where seizures, where most of our kids have seizures, but the seizure frequency is fairly random and is really dependent upon kids. I'm not sure what it is with Syngap1, but that may not be the best. But again, that may not be the best outcome measure to look at because of that, right? So if kids go through long sequences where they're not seizing and then have shorter sequences where they're seizing a lot, it can be difficult to time a clinical trial correctly, right? So, but in a good clinical trial, even if that is the main outcome measure, clinical trials will often have, or should have, different outcome measures that they look at, right? So it may not be the main outcome measure that the FDA is going to utilize to see whether a drug is effective, but there will be secondary measures that need to be looked at. And this is really where the natural history study and the disease concept models that you're developing come into play, because these are telling you how the disease is progressing. And if you're using measurements, let's say like that you may be familiar with the Vineland and the Baileys, and as standardized measurements that are utilized for development in kids, they're also utilized in neurodevelopmental disorders. And as you said, it's difficult to sometimes use those measurements as they were written, because Syngapians don't develop the same way as neurotypical children do. But those scales can be used if they are adjusted, or if you're using instead of the normal. Usually in Bailey's, you're comparing to the normal or to an age range. Instead of comparing to age range, you just look at the base number values, for example. You can kind of utilize some of those scales. But
sorry, so you're just comparing to yourself?
Yes.
You'd have each participant compared to self over time.
Yes.
Got it. Okay.
Yeah. And that's part of getting ready for clinical trial, right? Is to understand what outcome measures work within the population, right? So if seizures aren't going to be, seizures can work in some, and that becomes a measure, right? But also, how do we measure communication, for example? How do we measure movement? You know, so there are scales that look at different motor abilities or movement abilities, gross motor abilities, fine motor abilities. How do we gauge those? Yeah, and can we use those in our kids, right? And if we can use them, how do we need to adapt the scales that are already utilized, that the FDA already recognizes as usable scales to evaluate these particular domains in kids? How do we adapt those to Syngapians, right? To evaluate a drug, right? So that's work that needs to be done now, that is being done now, that's starting to be done now, right? So I know that, you know, starting the natural history study at CHOP, so they're running many of those measures, and that's kind of the things that they're looking at for.
Okay, so
yeah.
So let me interrupt you on that, and then you can keep on with your train of thought. You're saying there are some validated measures, but I know we're also other validated measures, other measures are in development and trying to be validated. So is the idea to get as many of those as we can into our patient, you know, sort of like baseline for as many patients as we can, and then sort of try to figure out which validated measures does the FDA have a list of things that it accepts, or is there, how do we, you know, do we like go lobby to say, this is a great measure for us, will you please accept it? Like, what's that process like?
Yeah, that's basically what you have to do. So the FDA does not have a list of measures that's acceptable for you know, neurodevelopmental delay disorders as a whole, right? Which would be great. Now, maybe they will in the future, but they don't at the moment. So basically what you have to do is you have to go and look within your population and utilize these outcome measures and say, hey, look, you know, if we take let's say the Baileys, and we say, let's take a look at, you know, one component of the Baileys, and you know, we adjust it, we either adjust the questions that we ask, or we adjust how we rate the kids, and we say, hey, look, now if we do this, if we take a look at our population, we see scoring that is distributed within our population. So that's important that you see the scoring is distributed, because that means that the scoring can increase, can get better, right? So if you're taking a measure where kids score 0, if they, you know, if you get a small improvement or an improvement, if they're so far below 0, you'll never get higher, right? So that's a basement effect. So you need to have evaluations or scales where your population of kids have a range, and then you can, the idea is you show that within a, you know, a certain number of the population. And unfortunately, there's no magic number where this works. It's just convincing the FDA with the data that you generate to say, hey, look, we've looked at 50, 100 kids with Syngap over this age range. We see this in this particular scale, we see that there is a range of values. And ideally, we see that it increases with age or something, or it has is associated with some other some biomarker or some other you know, another scale some way. But at any rate, you're convincing, you need to go eventually convince the FDA that this becomes a useful scale in our population. And that's what a lot of the neurodevelopmental disorders are doing now, right? So the disorders that we always talk about who are ahead of us, you know, Dravet syndrome, for example, you know, that's what they've done, is, you know, they have looked at, you know, different scales, what works in that population, and you try to go to the regulatory agencies and you say, this is what we have, and we think that this will work. And then it's basically up to the FDA will accept that. They'll say, okay, if you show us this changes, you can convince us that this becomes a valuable scoring or a valuable scale. So even a scale that are subjective scales, the FDA will accept. Clinical evaluation scale, just global assessment, right? So you see your pediatrician and the pediatrician says, oh, your child looks much better today than the last time I saw them 6 months ago, right? So that is something that the FDA can accept. I will say that the FDA tends to be less acceptable of global evaluation scores from caregivers than they are from providers.
Okay, and do they care that it's the same provider over and over, or it's any provider?
They do. I can't say that they don't care, but generally, I don't think that's a huge issue with them.
Okay.
You can make an argument that it's best to have it as the same provider because they're basing their global impression on the same thing. But on the same hand, if you want to be, if you have a true effect, that should be observable by any provider.
Right. Okay, thank you. Well, I want to go to the Q&A. Olga, who introduced you, has a question. Do you want to read it, or do you want me to read it out loud?
I have the.
So I'll just read it to the audience. With gene replacement therapy having potential to overexpress, do we know how overexpression can affect patients in general? And secondly, the effect of overexpression of Syngap1 protein.
Yeah. So that's an excellent question. And that's one of the reasons that I pointed out the fact that something like gene replacement therapy can cause overexpression. Because there are some proteins that are highly regulated in neurons or cells in general. And they're highly regulated because if they are overexpressed, they can actually have a negative effect on cells or neurons. For Syngap1, I don't know whether or not that's known. So, and the easiest way to do, that's something that would have to be preclinically determined by basically taking let's say an AAV that expresses Syngap1 and putting it into a mouse and just overexpressing and see if you can see any negative effects of doing that. Obviously, you're limited to what models are available. You can overexpress in neurons in a plate and see whether or not you see any large changes in other proteins or other genes or anything, and get an idea if you have any sort of negative effect on that. You can do animal models. You can do zebrafish or mice or flies or rats or whatever to see if there are any negative effects. You can look at non-human primates to look at that. But obviously there's no guarantee that it's going to be different in people, right? So that is always a concern with gene therapies, because those types of gene therapies, as I said, are permanent. So if there was a negative effect for a lot of types of gene replacement therapies, there's no way to get rid of that negative effect. So that is a very good question. But I would say for the vast majority of proteins and genes, there does not seem to be an issue with overexpression. So sometimes if the cell may have a way of getting rid of anything that's overexpressed that it doesn't want. And for others, it doesn't really matter. So.
Are there, I'm surprised I don't know this off the top of my head. Are there any diseases, monogenic disorders, neuro or not, for which an AAV gene replacement treatment is approved?
I
mean, I can imagine maybe a blood or liver one, but I don't know.
I want to say yes, but I'm not sure that it's for sale any longer. So one of the first gene therapies that were approved was, I believe, a gene replacement therapy. And I forget what this disorder was, but it was. I don't think it's no longer marketed because it never really sold. It was for I think it was an enzyme replacement therapy.
Right. So not a brain has a different solid organ that has more issues than blood. Okay. Anything I should be asking you that I haven't asked you?
No. So, but if we go back to the overexpression issue. So apart from gene replacement, the other thing that that you need to be aware of is that certain mutations in genes can cause what's called a dominant negative effect, which means the mutation, the problem with the mutation isn't that it makes a defective protein. The problem is that it makes a protein that is having a negative effect on the cell as a whole, right? So for a lot of monogenic diseases, we think of those diseases as being haploinsufficiencies, which means that you're just making half as much of the normal amount of protein, and so that's what's causing the disease. But for some mutations, the protein that's made, and these are usually going to be missense mutations, that protein is being made, but that protein actually has a negative effect. It's having a biological effect. That biological effect is a bad effect, though. And so it's actually worse to make more of that protein. So if you had a therapy that was designed to increase the expression of protein in general, and it could not, like an ASO, for example, that increased RNA, but it could not tell apart the mutated RNA from the unmutated RNA, right? From the good gene, the bad gene, it would make, it would increase the expression of both the good protein and the bad protein. So you would not have, so it wouldn't work. In fact, it may actually be worse because you might end up making more bad protein and have a more negative effect. So that is one of the reasons why, for example, it's important to understand how the variants, what the pathologic effect of the variants are, especially missense variants. I hope that made sense. I'm not sure it did.
Yeah. Well, I'd say the one thing I would add to that with dominant negative is a dominant negative is where the pathogenic variant is interfering directly with the wild type variant.
Yeah.
So, because you can also have something that just does something new and random, but isn't
true. Yeah.
Isn't its mode of action isn't through the wild type protein. So that's what dominant negative is. And it can be, yeah, it's really mystifying. So that's why we need good asset. That's why we're funding 4 different projects to look into what is going on with the missense mutants. And for that, we need structure, sort of structure and stability data. We need assay data. We need lots and lots of stuff. So, yeah, I guess the one question I always come away with is what are I think most of the trials, they have this period where they look at negative effects, right? And so with small molecules, I understand that. You can say, what are the side effects? Oh, we tried tried a few different anti-seizure meds, anti-epilepsy drugs. And one of them was like, oh yeah, the people who learned about Syngap, they think this is going to be the best one, the very best one. And so we try it, and his negative behaviors just go through the roof. Like nothing gets better, except that he gets incredibly aggressive. And people had thought, oh, that's the opposite of what we thought was going to happen. And so you wait a while, and then the drug washes out, and then you don't go back to it. And then in his chart, it says he's allergic to it. He's allergic to it because he had this bad reaction, right? That's how my medical chart reads for him. So, so I don't quite understand the safety stuff for the precision medicines. I guess for the ASOs, you can try a low dose. And so I guess there's dosing. I guess the gene replacement is the only one that I guess it's sort of mystifying to try to figure out what's the pathway towards finding out if it's safe.
Sure. Yeah. So so for a gene replacement therapy, you still do a dose. You're still dosing patients, but generally, how gene therapy trials work is that you start with a low dose, and for a gene therapy trial, the FDA requires that as being a dose that can have a positive effect.
Can or cannot?
Yeah, it has to be able to potentially have a positive effect.
Okay.
So it could be the lowest dose where you would
predict
your doses on animal studies, right? But so you take the lowest dose that you saw positive effect in animals, and that basically becomes your human dose in a clinical trial, adjusted for the size. There's a mathematical formula to adjust animal and human doses. So, but you use that as your low starting dose, and you basically treat a patient. Usually you start off with one patient, and you see, is there any immediate negative effect, right? Do they have an immediate bad reaction to that? And then you might dose a second patient. That second patient may be dosed a couple of weeks or a month later, because you're observing that first patient, and that's usually how this goes. And you dose 3 patients at a low dose, at the lowest dose, and then you follow them for a couple of months, sometimes up to 6 months. The FDA will decide on kind of what that time period is, and that's exactly why you're doing that. Is because you don't know, it's not going to wash out if there is a negative effect, and hopefully that negative effect, if there are adverse events from the gene therapy, they can be managed because they're some sort of systemic issue, for example. But yeah, it obviously it is a chance. You are rolling the dice with that, but for the most part, there really hasn't been huge issues. There have been some with AAV. Most of the serious events that have occurred are liver toxicity due to dose, and some toxicities associated with dorsal root ganglia, which are neuronal structures along the spine. And so, but those are toxicities that are known, and there are ways to try to get around some of those toxicities. And then, if you don't see any adverse events or serious adverse events, then you go up to the next dose.
Yeah. Okay, well, I feel like I could talk to you for a few more hours. It's 10:20. You may need to go. How are we doing?
I'm fine. Yeah.
Yeah. Okay. Well, I just wanted to mention that, for Spinraza, for ASO, Spinraza was the first one that I know of. And that's a a case where people are literally dying without it. And so that's a case where people want to try something, and it worked great. So that was really interesting. So I'm just wondering I mean, I'm just thinking out loud. I'm wondering, maybe for ALS, or, maybe there's some kind of brain disorder where people really, it's sort of a last chance. And maybe that's what will be first in the AAV gene replacement, and we'll get more understanding there. I don't know. That's what makes sense to me, because my child's not dying. I mean, he's got a ton of problems, and I'd love his life to be better. But making it a lot worse seems like, I went through really hard times with him, and we're on the other side of it. And it seems.
Oh, yes, that's absolutely something that people are going to have to balance when, these new therapies come along is, first do no harm, right? We want to try something to help our kids, but we don't want to make anything worse, right? And so that's a major concern with the FDA. And as you said, for a disorder like spinal muscle or SMA, the issue is, it's treatment, or you're treating kids who are going to die, right? And so that's an clinical outcome that's easy to measure, not dying, and
over the natural history of the disease. Yeah. You're like, typically 90% of patients have died by this age, and these patients are all alive past that age. So
yes, yeah. So in that case, it was kind of an easy decision, and with those are ASO drugs, and so they are, there had been a negative effect. ASOs will wash out because. And so that's one of the things that makes ASOs safer, obviously, which make them, and that's why a lot of companies are looking at ASOs, and why, those will probably be the types of precision therapies that will be quicker to be developed for Syngap.
Right.
But again, the ASOs might, work well, but, gene replacement may ultimately be a more permanent solution, right? So there are positives and negatives, and we just don't really have enough information to make a decision as to what's going to be the best way moving forward.
Right. So I guess our role at SRF is to figure out what are the things that we have to know for our own patients. And then who do we look to see who's doing stuff that we're not going to be doing, but we need to see what their results were. So I guess that's always our thing, is like, what's our little focus? What do we have to do? Because only we will do it, or only, it's really patient specific, and then keep abreast of all the, because we're obviously, every monogenic disorder wants to have a cure. So.
Right, yeah, and you're perfectly correct in the fact that by the time there's a gene replacement option for Syngap, something like that will have already been done in another disorder, and we'll have a much better idea about how that worked for specific neuronal disorder.
And maybe even to Olga's question too about expression levels, and there's lots of things that we could know more about.
Yeah.
I mean, and they'll be specific for each engineered construct, but yeah. Anyway, okay. Thank you so much, Jim.
You're welcome. So I hope people enjoyed it, or if they are watching at a later time, they enjoy it. And if anybody has any questions, I guess I should have put my email address up on one of the slides, but you can get me at I can give you my general email, which is jamesrgoss@outlook.com. So I'd be happy to answer any questions. I also have a Syngap Research Fund email address. So I think
Yeah,
that's,
yeah.
you know, here's Syngap.
Let's
Right.
see if I can put that up. Can I put it? I don't know how to do this. Is there a text? Yeah. Is it writing? There we go. So you said James R.
Goss at outlook.com.
There we go, peoples.
Yes.
You saw it here. Okay. Well, thanks so much, Jim.
Oh, you're welcome. Thank you.
Bye.
Bye.